Hello everyone. In this class, we'll learn about male reproductive system. A reproductive system is unique because it ensures the continuation of the species. The main difference from other systems like cardiovascular system, respiratory system is that other systems function for survival of the individual, but function of reproductive system is not necessary for survival of the individual, but it is for the continuation of the species. The organs of reproductive system can be divided into two portions, that is internal reproductive organs and external genital organs. Some of the abnormal sexual differentiation can occur, which could be because of the chromosomal abnormalities. Turner syndrome, where there will be only 45 chromosomes, it is called as 44XO syndrome, and it is also called as gonadal dysgenesis or ovarian dysgenesis. Here, gonads are rudimentary and they are absent. Usually, they will be having female external and internal genitalia. Stature will be short and puberty changes do not occur in them. Whereas in Klinefelter syndrome, where there will be presence of 47 chromosomes, that is 44 XXY. Here, genitalia of a normal male will be present, and testosterone secretion at puberty is normal, but seminiferous tubules are absent. Can see mental retardation in them. Whereas Down syndrome is autosomal dysfunction of chromosome 21, which produce trisomy, which is also called as Mongolism. Some of the symptoms here, they will be having broad face, epicanthus, hand with single transverse crease, and consistent infection that leads to death in them. Some of the hormonal abnormalities include female pseudohermaphroditism and male pseudohermaphroditism. In female pseudohermaphroditism, male genetic development occur in genetic female exposed to androgens from other source during 8th to 13th week of gestation. Whereas in male pseudohermaphroditism, there will be development of the female external genitalia in genetic males. Some of the causes for male pseudohermaphroditism are embryonic testes which are defective and androgen resistance. Bar bodies or sex chromatins. A bar body is highly coiled, genetically inactive X chromosome seen in the female, where other X chromosome is active. It can be detected in sex chromosome from buccal cells, vaginal mucous cells, and spinous epidermal cells. Number of bar bodies is equal to number of X chromosome minus 1. That is, if they have 2 X chromosomes, they will have 1 bar body, and if they have 3 X chromosomes, they will have 2 bar bodies. Coming to the male reproductive system, the reproductive functions of male can be divided into 3 major subdivisions, which includes spermatogenesis, that is nothing but formation of the sperm, performance of the male sexual act, and third one is the regulation of male reproductive functions by various hormones. Blood-testis barrier, the tight junctions between adjacent Sertoli cells near the basal lamina form blood-testis barrier. These creates a specialized immunological safe microenvironment for developing sperm. Steroids can penetrate this barrier with ease. Major germ cells pass through this barrier as they move to the lumen through the formation and breakdown of these junctions. Functions of blood-testis barrier prevents large molecules from passing from interstitial tissue and the part of tubule near the basal lamina. They are also important for maintenance of composition of fluid in the lumen of seminiferous tubules, like that is, they maintain little protein glucose but rich in androgens, estrogen, potassium, inositol, glutamic, and aspartic acids. So, which eventually depends on the presence of blood-testis barrier. Blood-testis barrier protect the germ cell from the blood-borne noxious agent, and it also helps in to establish an osmotic gradient that facilitates movement of fluid into the tubular lumen. This structure showing the blood-testis barrier, where they are made up of Sertoli cells and mainly the tight junctions between the Sertoli cells and thick basal lamina. And you can see the Sertoli cells harboring the different stages of spermatic cells. Coming to spermatogenesis. Spermatogenesis is a process by which new spermatozoa are created from Spermatogonia is called as spermatogenesis. In human, it takes on an average 74 days. The estimated number of spermatids formed from single Spermatogonia is 512. Phases of spermatogenesis. The first phase is phase of mitotic division of Spermatogonia. Here, Spermatogonia divides mitotically 7 times to form 128 Spermatogonia. The second phase is phase of formation of primary spermatocyte by maturation. 128 Spermatogonia undergo maturation to form 128 primary spermatocytes. They are large cells with a diploid number of chromosomes. The third phase is phase of formation of secondary spermatocyte by first meiotic division. During first meiotic division, that is reductional division, 128 primary spermatocyte is converted into 256 secondary spermatocytes with a haploid number of chromosomes. Fourth stage is phase of formation of spermatid. Each secondary spermatocyte undergoes second meiotic division here, similar to the mitosis, to give rise to spermatids. Thus, the total of 512 spermatids is formed having haploid number of chromosomes. Fifth phase is formation of the spermatozoa. Spermatids do not divide further but undergoes morphological changes to form sperms or spermatozoa. Spermiogenesis is conversion of spermatids into the sperms. This process takes place within cytoplasm of the Sertoli cells and depends on the presence of androgens. So these are the different stages of spermatogenesis. Primordial germ cell, they remain quiescent till the puberty, and after puberty they starts dividing by mitosis, and then they will form the primary spermatocytes. The primary spermatocytes by meiotic division 1 they get differentiated and converted into mature sperm. Factors that are influencing spermatogenesis. Testosterone. In high concentration, the testosterone is essential for growth and division of testicular germinal cells, which is the first stage in forming sperm. Luteinizing hormone, it is secreted by anterior pituitary gland, helps in stimulating Leydig cells to produce testosterone, which is essential for spermatogenesis. Follicular stimulating hormone is a key gonadotropin for spermatogenesis. It is secreted by anterior pituitary. It helps in the conversion of spermatid to sperm by stimulating Sertoli cells to produce some local factors. Growth hormone is necessary for controlling the background metabolic functions of the testis. It especially promotes the early division of spermatogonia themselves. Then estrogen is formed from the testosterone by the Sertoli cells when they are stimulated by follicular stimulating hormone. They are also essential for spermatogenesis. Sertoli cells, which secretes androgen binding factors or proteins, inhibin and Mullerian inhibiting substance. They do not synthesize androgens but contain aromatase, the enzyme responsible for conversion of androgens to estrogens, which helps in the process of spermatogenesis. Androgen binding protein helps to maintain high stable supply of androgens in the tubular fluid. Inhibin inhibits FSH secretion, and Mullerian inhibiting substance causes regression of the Mullerian duct in male during fetal life. The seminal fluid with fructose content help in the maturation of the sperms. Vitamin E, C and several members of B complex are required for normal spermatogenesis. The general factors which affect spermatogenesis are temperature, where optimum temperature at around 32 degrees Celsius is maintained for the process of spermatogenesis. Radiation sometimes can degenerate seminiferous tubules. vasectomy, and age are the other factors which are going to affect the spermatogenesis. As the age progresses, the spermatogenesis regresses. Some of the hormones which are necessary during the different stages of spermatogenesis. During the stage of proliferation, follicular stimulating hormone and growth hormone plays important role. During the stage of growth, testosterone and growth hormone plays important role. At the stage of maturation, testosterone and growth hormone, whereas during stage of transformation, testosterone and estrogen, they play an important role. Spermatozoa. The sperm is about 60 millimeter long and is a long single cell which has a head and a tail. The head is the nucleus of the cell containing DNA material. Head is capped by acrosome made up of mucopolysaccharide and acid phosphatase enzyme. The acrosome is covered by a membrane called as acrosomal membrane. In the fallopian tube, acrosomal membrane is removed so that the acrosomal enzymes are exposed. These enzymes are digestive in nature that facilitates their activity by enabling the sperm to penetrate the ovum to cause fertilization. The removal of acrosomal membrane is called as sperm capacitance. The highly elongated structure of sperm enable it to swim forward in the female genital tract at the speed of 3 millimeters per minute. Sperm reach the uterine tube 30 to 60 minutes after the copulation. So this is the structure of the sperm, which has a head, neck, middle piece and the principal piece. And the head has a nucleus with acrosomal cap, whereas middle piece has abundant mitochondria. Semen. Semen is the fluid that is ejaculated at the time of orgasm. It contains sperms and the secretion of seminal vesicles, prostate, Cowper's gland and urethral glands. You see the composition of the semen. These are some of the substances secreted from the different structures. Like seminal vesicles, it produces phosphorylcholine, ergothionein, ascorbic acid, flavins and prostaglandins, whereas prostate gland produces spermine, citric acid, cholesterol, phospholipids, fibrinolysins, fibrinogenase, zinc and acid phosphatase. And they have a plenty of buffers like bicarbonate and hyaluronase. The specific gravity of the semen is 1.028 and pH is between 7.35 to 7.5. The average volume per ejaculate will be around 2.5 to 3.5 ml. Sperm count is around 80 to 120 million per ml, with fewer than 20% of the abnormal forms. Seminal vesicles contributes to almost around 60% of the total volume of the semen, and prostate gland around 30%, 20 to 30%. Note that clotting enzyme from the prostatic fluid causes the fibrinogen of the seminal vesicle fluid to form a weak fibrin coagulum that hold the semen in the deeper regions of the vagina. The coagulum then dissolves during next 15 to 30 minutes because of the lysis by fibrinolysin formed from the prostatic profibrinolysin. Nutrition to sperm is mainly provided by the fructose, and fertilization is enhanced by the prostaglandin present in the seminal vesicles. Coming to Sertoli cells. Sertoli cells are large, complex glycogen-containing cells that stretch from the basal lamina of the tubule to the lumen. Some of the functions of Sertoli cells are: Sertoli cells secrete androgen-binding protein, inhibin, activin, and Mullerian inhibiting substance, that is MIS. They have receptor for the follicular stimulating hormone and testosterone. After combining with FSH, Sertoli cells stimulate the first half of the spermatogenesis. Subsequently, testosterone Sertoli cells binding causes development of the last half of the spermatogenesis. Sertoli cells are components of blood-testis barrier, and Sertoli cells they produce estradiol from testosterone. They also provide nutrition to the germ cells, and they also help in conversion of testosterone into 5-alpha dihydrotestosterone. Sertoli cells and epithelium of the epididymis secrete a special nutrient fluid that is ejaculated along with the sperm. Testosterone. Testosterone, the principal hormone of the testis, is a C19 steroid with the OH group in the 17th position. Synthesis of testosterone occurs from the cholesterol in the Leydig cells and is also formed from the androstenedione secreted by the adrenal cortex. Testosterone secretion occurs at the rate of 4 to 9 milligrams per day in normal adult males. Small amount of testosterone are also secreted in females. The secretion is under the control of luteinizing hormone. Almost 98% of the testosterone in the plasma is bound to plasma protein, and 65% is bound to beta globulin called gonadal steroid binding globulin or sex binding globulin, and 33% is bound to albumin. Mechanism of action of testosterone. Testosterone has several target cells. In the target cells of prostate, seminal vesicles, epididymis, penis, and skin, testosterone crosses the cell membrane and is converted to dihydrotestosterone by an enzyme 5-alpha reductase. In these cells, the dihydrotestosterone is the final hormone, and testosterone in these cells merely a prohormone. In the hypothalamus, muscle mass, anti-repituitary, testosterone is the final active agent and no dihydrotestosterone is formed. The androgen, either dihydrotestosterone or testosterone itself, depending on the tissue, combines with its receptor and the receptor loses its heat shock protein. Now the androgen receptor complex moves to the target chromosome and various proteins are synthesized and acts on target cells. Their actions on the sex organ or secondary sex characteristics and sex behavior in the fetus. Testosterone is formed in the 7th week of intrauterine life and it is responsible for the sex differentiation in male fetus. Exposure of some part of brain to the testosterone in the fetal life determines the sexual behavior in adult life. In the puberty, testosterone causes growth of external genitalia and accessory sex organs in the pubertal stage. Testosterone also causes appearance of secondary sexual characters in boys, that is, appearance of the beard, mustache, hairs on the chest and abdomen, as well as axillary and pubic hairs. The linear growth, muscular growth, deepening of the voice, and male psyche are also caused by testosterone. In adults, it is important for maintenance for accessory sex organs and it is important for development of the sebaceous glands and acne vulgaris. It is necessary for spermatogenesis and sperm motility. On the metabolism, it has mainly the anabolic effects. On the proteins, it increases the synthesis and increases the breakdown of the and decrease the breakdown of the protein, leading to an increase in the rate of growth. And in the bone, it causes epiphysis to fuse with long bone, and it also increases the growth. It also favors the calcium deposition, and it also promotes the growth of the muscle mass, and muscle mass gets doubled. It also has action on the electrolytes where it causes retention of sodium, potassium, calcium, water, phosphate, and sulfates. Androgenic drugs like nandrolone are used as anabolic steroids. These agents are not easily converted to dihydrotestosterone, so they have a minimal action on sex organs, but they act on muscles to increase the muscle bulk and strength. Unscrupulous sports persons misuse this drug to increase their muscular efficiency and it is called as doping. Coming to the regulation of secretion of testosterone. Secretion of testosterone from Leydig cells during fetal stage is caused by human chorionic gonadotropin from placenta. After birth, its secretion is absent till adolescence, and during adolescence, it starts again under the influence of gonadotropin releasing hormone. So, gonadotropin releasing hormone leads to the secretion of LH from the anterior pituitary. LH is going to act on the interstitial cells of Leydig to produce testosterone, and the plasma levels of testosterone, by negative feedback, it reduces the secretion of LH and GnRH from the hypothalamus. Some of the applied aspects related to male reproductive system. Cryptorchidism. The testis develops in the abdominal cavity and migrate to the scrotum during fetal development in the 7th week. The failure of descent of testis cause testis to remain in the abdominal cavity or inguinal canal, leading to sterility, called as cryptorchidism. Testicular feminizing syndrome. It occurs due to the congenital lack of androgen receptors. Testis and testosterone secretion is normal, but testosterone cannot act due to the lack of receptors. So there is development of female genitalia and there is no enlargement of breast. Some of the tests which are done to test the male infertility. Semen analysis. Here the masturbation sample of semen is obtained and examined for sperm morphology, motility and count. Normally the volume of an ejaculate is 2 to 6 ml. If analyzed within an hour of ejaculation, more than 60% of the sperm should be normal in morphology, more than 60% of the sperm should be motile, and the sperm number in the ejaculate should be more than 60 million. At a testicular level, the function may be assessed by measuring the testosterone levels, that is free and protein-bound testosterone. Sex hormone binding globulin may be measured in abnormal testosterone levels. So this is in brief about male reproductive system. Thank you.